I'm pleased to welcome you to our presentation today as part of the ongoing 2023 CCOVI lecture series. This afternoon, we are excited to welcome Dr. Justin Renkema, research scientist in entomology with Agriculture and Agri-Food Canada and Vineland, Ontario, and CCOVI professional affiliate. Justin has a PhD from Dalhousie University, was a postdoctoral researcher at the University of Guelph, and was an assistant professor of entomology at the University of Florida, with a research and extension program focused on pest management in strawberries and blueberries. Justin's research program focuses on developing integrated pest management strategies in grapes, tree fruit, and dairy crops. He is currently working on spotted wing Drosophila in berries, cyclamen mite and weevils in strawberries, leafhoppers and insect vectors of Grapevine Red Blotch Virus in vineyards, and oriental fruit moth, other leaf roller moths, and ambrosia beetles in tree fruit. Please join me in welcoming Justin.
All right, thank you for the introduction, and it's a pleasure to be here today and to see everybody in the room and those of you who are online. And so the subject of my talk today is going to be work that we've been doing for the past 3 or 4 years on trying to identify which insect species are vectors of this really important fairly new virus in grapes. But by way of a little bit of a further introduction, before we get into that, I thought I would just put one slide up that shows some of the other projects that I am currently working on or have worked on or will be working on that involve grapes. So just kind of briefly for now, but if you have more questions about any of these projects, feel free to ask at the end. So with Tom Lowry, who is located in BC, we've been looking at leafhopper biocontrol with parasitic wasps for a few years here in Ontario, looking at different dynamics between different species of leafhoppers, the Erythronura species, and their parasitic wasps, which are sometimes called fairy flies, but they're in a genus called Anagris, and looking at which species of which are interacting. Sometimes it's hard to determine because the Anagris are egg parasitoids of the leafhoppers. We've also been looking at which are the overwintering plant hosts of Anagris, so they don't stay in the vineyard during the winter, they parasitize other leafhoppers on other woody plants outside of the vineyard. So if we know which plants might be beneficial for increasing the populations of these parasitoids, that would be a helpful management tool. So that project is almost wrapping up. We have a lot of data to go through at this point. Currently, we've been doing some work in the lab this winter, looking at the ability of using UVC light to manage grape leafhoppers. And this is in conjunction with Wendy McFadden-Smith. And we're trying to target the nymphs of the leafhoppers because they are smaller and more susceptible to UVC perhaps than the adults. And just doing this in terms of looking at, does the UVC kill them? Are there non-lethal effects on their development, etc. And then finally, a project that will be starting this summer is with Ian Willick, who's a scientist in Nova Scotia. And this is looking at how this silicate-containing compound can perhaps reduce foliar feeding by pests, and specifically caterpillars in grapes. But now to return to the main topic, Grapevine Red Blotch Virus. I think in the CCOVI lecture series this winter, there's going to be some other talks that cover different aspects of this virus. So in terms of what it is and what it does itself, I'll just kind of go over that briefly. It's relatively new within the past 10 or so years that it's been documented to have some significant effects in vineyards, specifically and mainly in North America. It produces these foliar symptoms that on red berry cultivars are red blotches, hence the name, that tend to coalesce over time. In the white berry cultivars, the chlorotic areas aren't necessarily red, but they kind of become chlorotic and then necrotic, so that tissue dies off. We often see progression of symptoms throughout the season from the older to the newer leaves, and we get premature leaf drop. Fruit symptoms include delays in ripening, smaller berries, juice chemistry is different, so the quality of the wine is affected, and typical yield reductions. So we know that this virus can spread kind of long distances if plant material is infected. So clean plant material is key to combating and managing this virus. But it appears that also there is some secondary or local spread, usually within vineyards. And what is contributing to that spread? Well, it's probably due to insects. So, I mean, a lot of the evidence for GRBV spread comes out of California vineyards, and in California it is definitely spreading. But what about in other places? How about Ontario? This is some data that Wendy McFadden-Smith has put together, just two different areas, two different blocks of vineyards, one Chardonnay and one Vidal, and you can see the Chardonnay over time, the percent GRBV increase is quite minimal, quite small. Perhaps it's not spreading in this vineyard, at least not very much. Whereas in the Vidal, it's a little bit of a different story, higher incidence and a larger amount of spread over the years that these vines were studied. And each block here, I believe, is an individual vine. So every vine here was sampled. So what about other regions? Some data from Jose, who you'll hear from next week, and Tom Lowry have looked at infection in a BC vineyard. And in general, they conclude there's a pretty low rate of spread in BC. And this study was looking at roguing, so removing plants that were infected. And so in 2018, here's the infection rate in this vineyard. All these infected plants were removed. And by 2020, the rate of infection is just 0.3%. And you can see there's only a handful of newly infected vines in those years after roguing. But all those new infections seem to be right beside a previously infected vine, which is interesting. And I'll get back to that a little bit later. In some other regions outside of California, there seems to be a lack of spread or any significant spread in New York vineyards. And also recently, papers have come out saying there's very low incidence in some of the European vineyards, and especially France and Switzerland, where these two studies took place. So the spread that we are seeing here and in some other places is most likely due to insects. And in order for the insect to spread the virus, it first has to have the ability to acquire the virus. So the first kind of part of my presentation is going to look at GRBV acquisition by insects. So we know this virus is a Gemini virus, and all Gemini viruses that we know so far in plants are spread by true bugs in the order Hemiptera. And this Gemini virus, we're pretty sure will be transmitted by and acquired by these leaf hoppers, plant hoppers, and tree hoppers, which is a subset of the Hemiptera order. So back, quite a few years now, there were some studies that were done in California vineyards to identify which insects were positive for the virus. So they went out with sticky cards, caught insects, identified them, and then tested at least a subset of them for the virus. And here are the results, kind of putting it together from a couple papers. But over 2 years in one vineyard, so this is the percent positive of these insects. So this is kind of candidate number 1, Spicistolus festinus, which is, you can call it the three-cornered alfalfa tree hopper. It's this one. And these are 2 different vineyards in these 2 different years, but 50 and 40% were positive. And I believe quite a few of them were captured in the vineyard. But there are some other species and genera where the percentages are relatively high, 40 to 70% or so. And then a couple other lower percentages of things that were identified to genus. And one vineyard was looked at for 2 years in New York, and I believe it was in the eastern part of the state, where basically the species that were found in California were not found in that New York vineyard, especially the alfalfa tree hopper, but some other different genera and species were found, but the percentages are fairly low. And we're going to get back to Stictocephala genus. That's the buffalo tree hopper. But in their study, they only captured 3 of them over 2 years, and none of them were positive. So this group that has published these studies is not the only group who have been looking at insects that are positive for the virus. This is another group in California who's looked at 5 different vineyards over 2 years, and they generally see, they captured some of the same insects. Top of the list is this alfalfa treehopper again. In general, their percent positive is a little bit lower than the previous studies, but you can see some higher percentages for some of these other genera. But some of them, the percentages are high, but the numbers that they've captured are quite low. And there's another recent study from Missouri looking at a bunch of vineyards and the surrounding vegetation. Here you can see in general that there's a lot of zeros in terms of GRBV positive insects. There is this one Entilia carinata that has varying percentages positive, and the percentages are varying in this case because they sometimes grouped individuals into a sample for testing. And this Entilia carinata is also a type of treehopper. It's the one here in the middle with this big characteristic thorax. So these other regions, we're finding insects that have the virus. What about Ontario? That was our initial question. And we set out basically trying to do more or less the same sort of study that has been done with sticky cards. So putting sticky cards out in a bunch of vineyards like this. We did in 2019 and 2020. We replaced them weekly, pretty much. And in each vineyard, we had kind of three sticky cards at 6 different sites for a total of 18 cards. And 3 of those sites were on a vineyard edge. This is just like one example. 3 of them we put in the interior. So we brought those cards back to the lab every week. We removed any of these hoppers that we thought might have the virus. We tried to identify them to family or genus and sometimes to species. A lot of them are hard to identify. And then we went through the process of extracting the DNA from a subset of them and testing them for GRBV. And here's kind of a big table of the results. I mean, don't worry about all this list of names. Basically, we found, as with, say, the study in Missouri, a lot of zeros, a lot of things that we tested were not positive. And the only insect that was kind of consistently positive at a relatively high rate was something called Melanolieris, and you saw that name on one of the previous lists. There's not a common name for this, so we're going to have to go by the genus name here for the rest of the talk. We've had some of them identified by the taxonomists in Ottawa, and they are this species Aridus, but basically the males have to be identified. It's almost impossible to identify the females, so I'm just going to call them Melanolieris for this talk. This buffalo treehopper, which is very similar to the alfalfa treehopper in California, so we're very interested in it. But in our study, basically we found very few of them actually in the vineyard on sticky cards, and none of them were positive. There was one genus here in the middle, Scaphoideus, where, out of all the ones we tested, it's pretty common. One tested positive, so that genus has been tested by previous groups, and there were a few positives as well. But it's probably not a candidate vector species. So for this Melanolieris, it's a plant hopper. If you try to find out, what's been published about it, very, very little. It's hard to really tell what, with anything about its biology or ecology. Kind of the genus and the family in general. Those adults can occur on a lot of different crops, but they've never really been recorded in grapes as far as I can tell. But none of them are necessarily known pests on those crops. They show up, they may feed. But one interesting thing, and we'll get back to this near the end of the talk, is that we know that the nymphs are subterranean. They live in the soil, so the adults lay the eggs in the soil. The nymphs develop by feeding on roots of different plants. And when they're ready to become an adult, they emerge from the soil. And so that could be an important characteristic if we're looking at this insect as a potential vector of the virus. So with all these Melanolieris that we captured and tested, and a bunch of them are positive, we could look a little bit at, like, does the positive rate in this insect vary throughout the season? So I put here kind of this is for 2020, where we captured more of them. In 2019, the numbers weren't as high. So for 2020, we started capturing them kind of near the end of July, and it was kind of 2 peaks almost. We captured very few for about a week here in kind of the middle of August. And so the blue is the total captures, and then The orange lines are the ones that have tested positive. So I'd say, like, to answer this question, you know, we've got positive ones right away here at the beginning of the season around the 50 to 40% range, and it's about the same later on. So there doesn't seem to be much variation. It's not like they were all positive near the end of the season and started out negative, the ones we captured at the beginning of the season. And then I also separated them out, you know, depending on where they captured. If we caught them on traps that were near the edge of the field versus the interior of the field, and we separated our capture period into these early and late, you know, is anything different? I mean, there's kind of some trends here. I mean, I haven't analyzed this with stats, but, you know, the positive rates are going up a little bit as we go from early to late here, which is a little bit reflected in the previous graph. And there seems to be more of these insects captured on the edge than the interior, but the percent positives on the edges are a little bit lower than the interior. So there could be something going on here with the dynamics of, you know, where this insect is coming from. When it gets in a vineyard, where is it feeding and where is it moving within the vineyard? In 2019, actually all the Melanoleris that we captured were from the latter half of the season, and the trend is a little bit opposite of 2020, as you can see, where we have higher captures in the interior, lower captures a bit on the edge, but a higher percentage of them testing positive. So, you know, it's hard to say, you know, I think, or to definitively answer this question at this point. So then fast forward a couple of years to this past summer, where we thought, instead of looking in the vineyards, let's look around some of these vineyards in the vegetation. Are we going to find these two insects, the buffalo treehopper and the Melanoleris, and how abundant are they there? So we selected 9 vineyards. Most of them are ones that we had sampled previously in the vineyard. And we went with sweep nets to vegetation every week, just about, from early July to early September. And we selected these 20-meter areas. We had anywhere from 4 to 6 per vineyard, and we did 20 sweeps through the vegetation in those 20 meters. We caught a lot of different things. We decided just to focus and pick out, you know, the species that were of interest and sorted and saved them all. And so currently we're working on extracting the DNA from them and testing it for GRBV. And because this buffalo treehopper genus Stictocephalus, some of them are quite hard to identify, we're going to hopefully barcode a subset of the individuals. So in terms of just their populations and where they are, you know, this is what we found. Buffalo treehopper, the green line, you can see from early July to early September, increases in abundance quite nicely, and then it drops off from about the 2nd or 3rd week of August. And it's relatively abundant in the vegetation compared to Melanoleris. You can see the black line is close to or at 0 for a lot of the weeks. And then I thought I'd separate the data just by the different vineyards. You can see that buffalo treehopper is everywhere. We can find it next to every vineyard we went to, and you know, there's a few more some places than others, but it's pretty much everywhere. Melanoleris is a bit of a different story, totally absent from 4 of the 9 vineyards, and then these other 5, I mean, you can hardly see the black bars. There's so few compared to the buffalo treehopper. So you can see the totals are quite different. And maybe 2 populations peaks, which would kind of correspond to those graphs that I previously showed you from the previous years on sticky cards. So that's something interesting to look into in the future as well. And so this is kind of like some preliminary data here. I said we're still working on testing them. So for buffalo treehopper, here's the numbers per week. Dark lines, or the darker green is everything we've tested that has been negative. I put a few gray bars here where, you know, you do run a PCR and you're looking for bands and it's really faint, and so we're basically retesting those. But I think, you know, upon retesting, it looks like they're probably negative. So far we haven't really found anything positive, but we do have a lot more individuals to test. Here's with Melanoleris, same thing. We pretty much tested them all. There's just 3 individuals from this week we haven't tested yet, and this maybe is a probably not at this point. So, I mean, just to summarize what I just said. So, yeah, we've got to test the rest of them, barcode, hopefully the majority of the tree hoppers, because it would be nice to know exactly what species we're working with. I think they're all, or probably 95% of them are one species, but there's probably a little bit of diversity there. And one thing we didn't do this past season is kind of categorize or identify any of the plants where we did the sweeping, so we could have some comparison of, oh, we caught a lot of buffalo treehoppers in this spot because it has these species of plants versus somewhere else. So we can go back to those spots again next year and do some of the plant identification. So just kind of to summarize again, on our sticky cards, about 50% of the Melanolaris have been GRBV positive. So is it a good vector candidate? You know, some literature says 30%, maybe 40% should be positive to be considered a vector. So we're at that range where we should continue working with this and test it for transmission. You know, one thing would be interesting to find out, are grapes feeding and reproductive hosts of this plant hopper? You know, it looks like it has GRBV and it likely is acquiring it in the vineyard. You know, nothing that we've captured outside the vineyard has it. And it's relatively low in abundance outside the vineyard, and I would say kind of low to moderate abundance in vineyards. You know, so far, really no buffalo treehoppers captured on vineyards, and probably none, or it looks like very few that we've collected in sweep nets around vineyards, maybe GRBV positive. So is it a good vector candidate? I know if we go by percent positive, maybe not really. It's also very rare in vineyards, but it's really abundant and common in vegetation around vineyards. So it's there, but is it going? It doesn't seem to want to go into the vineyard or have any reason to. But it is very closely related to alfalfa treehopper. And you know, we've seen it. We have visual evidence that it will girdle and feed grapes. So it can feed on grapes. So we will move kind of on to now transmission. So looking at these insects that can or may acquire GRBV, now can they transmit that virus to a clean vine? So just to kind of go over some of the literature on this, and we've already talked about three-cornered alfalfa treehopper. The first paper came out looking at an assay that was done in the greenhouse from an infected vine to a clean vine. And they put the alfalfa treehoppers on the infected vine for 48 hours to acquire it. Then they put the treehoppers on a clean vine to inoculate it. And then they tested those vines, and over time, only up to 3 out of the 15 vines ever became GRBV positive. So a fairly low rate, but it does seem like this species can vector GRBV. Some work was done elsewhere, like in Oregon, looking at some different species that occur there. I mean, this treehopper occurs in Oregon as well. But they really had really low rates of any transmission. They really couldn't even replicate this 20% from this Bader 2016 paper. So, and then, well, about 2 years, a year and a half or so now, a paper came out of Mark Fuchs's lab at Cornell that really has, I think, clarified a lot about three-cornered alfalfa treehopper and its ability to vector this virus. So I'm just going to take you through some of the images and work that they have done. So they kind of tried to replicate what was done before. So 48 hours on infected grapes, 48 hours on uninfected grapes. They could not get any of those uninfected grapes to become GRBV positive, even though most of the treehoppers that they tested were positive. So this is alfalfa treehopper. And they have a laboratory colony of this species at Cornell, so it's, you know, they can do a lot of replicates and work with a lot of insects. So what they thought of doing as well, I think they, you know, we had an idea that this is a circulative virus, so it's got to move through the insect to be passed on to the plant. And that really means once the insect feeds on an infected plant, it goes into the gut, then it has to pass into the hemolymph, or like the blood of the insect, and then it has to get back to the mouth parts and the salivary glands so it can be injected into the plant. So they dissected a number of these insects. You know, 100% of the guts were GRBV positive, but none of the salivary glands were at 0 to 13 from these insects that only were on an infected vine for 48 hours. So it seems, you know, logical then that none of the uninfected vines became positive. So what they decided to do is run some experiments where they increased the acquisition period to 14 days. So give these insects a long time to feed on the vines, for the virus to pass through the hemolymph and back into the salivary glands. And they also did this kind of gut clearing period on alfalfa, which is a non-host of GRBV, for the insect to have some time to rid any of the GRBV out of its gut. And so in that case, so with the gut clearing, around half or so of the tree hoppers were GRBV positive. And then hopefully that they're positive because at that point, the virus has moved into their salivary glands as well. And if there's no gut clearing, a higher percentage were positive, but it's likely they were only positive because it was still in the gut. So then kind of the ultimate experience here then, or the experiment, or they did some other ones, but this is the one that we tried to replicate here, is take an infected grapevine, give them a long acquisition period, 15 days, and they basically determined that 10-day acquisition period was the minimum amount for GRBV to get into the salivary glands. And they had still only a 20% positive rate though. So 15 days, 2 days on alfalfa, and then instead of trying to transmit to another whole plant, they tried to get the insects to feed on an excised grape leaf and transmit the virus to that leaf. And they gave them 4 days to do that. And still in that case, only 4 out of 39 leaves were ever GRBV positive, 10%. So we're still pretty low, even though all these insects should have had enough time if they fed on the virus to get it into their salivary glands. They're still not transmitting it at a really efficient rate. A couple other things they did in this paper as well, which are interesting, is that they found that the GRBV can be transmitted from the nymphs to the adults. So if nymphs fed on infected tissue, and then once they become adults, the adults still had the virus. And the titer decreases over a fairly long period of time after feeding. So GRBV is probably a non-propagative virus. That means it doesn't increase in titer over time in an insect. So kind of in conclusion, this paper showed that, yes, this tree hopper is a vector of GRBV, but it's probably not an efficient vector. It can transmit it, but it doesn't do so very well, essentially. There's another recent paper out of Mark Fuchs's lab as well that, among some other things, looked at how well can this tree hopper transmit the virus between cultivated and free-living vines. And interestingly, you can see that regardless of if the donor vine is cultivated or free-living, if the recipient vine is free-living, those transmission rates are quite high. So it looks like, compared to much lower rates for cultivated vines. So there's something going on here as well that would be interesting to explore further. And they're using free-living vines that came from California.
Sorry.
Yeah.
Free-living.
So I think these are vines that they found in riparian areas. Wild, yeah. Okay, so moving on to the work that we've done here in Ontario. First, we tried some transmission assays in 2021. So because we haven't been able to rear either of these species, we went with sweep nets in vegetation around vineyards, and we collected as many as we could. We brought them into the lab into cages with GRBV positive vines, and we kind of held them en masse on those vines for 48 hours, 2 days, to hopefully acquire the virus. Then we moved them in groups of 5 to an individual vine in another cage for 2 days, and this is a cap rank vine that's been tested as GRBV negative. And we had lots of replicates with the buffalo treehopper, fewer replicates with our Melanoliaris. So then after 2 days, we took out the insects, we froze them, extracted DNA and tested them. We let the vines grow to hopefully, if they had GRBV, to let the titer increase. And we sampled 2 leaves per vine with the petioles 5 months after the experiment and also 14 months. So as you might expect from the experiments with three-cornered alfalfa tree hopper, since we only had a 2-day acquisition and 2-day inoculation, at least with the tree hoppers, we did not find any of the vines GRBV positive. They were all GRBV negative. And also very few of the insects that were used tested GRBV positive. So each plant here, 14 is a replicate. There were 5 per plant for a total of only 4 of 70 or 6% GRBV positive. So probably numbers too low to really transmit the virus. With the Melanoliaris, we also did not get any positive vines, so it doesn't look like there's any evidence here of inoculation. But differently, and what we would expect based on our field results, 74% of these insects after they fed on an infected vine for 2 days were positive. So they can acquire it, but so far we haven't been able to show that they can inoculate it. But now that what we know about this long acquisition period, the virus needs time to get from the gut into the salivary glands. Basically, we maybe just have not given these insects enough time. So in 2022, we ran another transmission assay and we changed our methods a little bit. Again, we went out and just collected insects. In 2022, we only worked with buffalo tree hoppers. It was just very hard this past year to find enough of the Melanoliaris to work with. Instead of putting them on a whole plant, we confine them in sleeves on about 10 to 12 leaves of an infected vine. That way they're more confined and hopefully they would feed. And we left them for a much longer acquisition period, as that paper showed is needed for alfalfa tree hoppers, so anywhere from 12 to 14 days. We did a 2-day gut clearing period on alfalfa, and then we used an excised leaf and a 4-day inoculation. And we basically did the same thing after those 4 days. We froze the petiole and tested it for GRBV, froze the insects. And with all these insects, we decided to, when we took them out to extract the DNA, we dissected them and removed the head from the body and tested them separately, with the salivary glands being in the head. And in terms of, we ended up with 6 replicates, and after 12 to 14 days on those leaves, and 2 days on alfalfa, we didn't have great survival of buffalo tree hopper. Like, grapes is probably not a great feeding host. So some replicates, nothing survived, and the best survival was only 50%. So then after 4 days on the leaf, we got pretty good survival of the buffalo tree hopper on that leaf, except for 1 replicate where nothing survived. So the results. So I'm just going to show, this is one gel image from one of the replicates where we had 5 buffalo tree hopper on that excised leaf. And what you're looking at here is basically this side of the gel is one set of primers and this side of the gel is another set of primers, so they're showing the same thing. The odd numbers are the bodies, or the even numbers are the bodies, sorry, the odd numbers are the heads. And we're looking for a band that's, in this case, about 257 base pairs, so right here, and you can see that all the bodies, there's a bit of, pretty much have a band in the expected size, but none of the heads do. And the same is true for the other set of primers. So even though we gave this insect a long acquisition period, at least out of 5 insects, we couldn't find it in the salivary glands. And as you would expect then, the recipient leaf did not test positive for GRBV. Even though we did see evidence of girdling on the petiole of this leaf when those 5 tree hoppers fed on it, so they were feeding on it. I didn't get a good picture of the girdling, but this is, I pulled this one off the internet. This is the girdling from alfalfa tree hopper. It looks very similar to what buffalo tree hopper does. So to summarize basically our transmission work the last couple seasons, we don't have any, we haven't been able to show that GRBV is definitely transmissible by either of these 2 insects yet, even though I think we're getting there. So in 2021, basically we didn't run the experiment for long enough. There was no GRBV in the insect salivary glands after 4 days. Everybody's shown you that in the alfalfa tree hopper you need at least 10 days, so it's that's probably the reason. In 2022 with buffalo treehopper, there still seem to be no GRBV in the salivary glands, but we're only basing this on 5 individuals that we've tested so far, so we may need more. Even with alfalfa treehopper, even with the same protocol, they're only getting 10% vine to leaf transmission. So these insects are not so efficient at moving the virus from their guts to their hemolymph to their salivary glands. But there could be also some differences, of course, between alfalfa treehopper and buffalo treehopper physiology, biology, and the ability of the virus to move from the gut into the hemolymph and then into the salivary glands again. So kind of next steps with buffalo treehopper, even though we haven't been able to show transmission yet, there still is a good possibility that this species could be an inefficient vector of GRBV, very similar to alfalfa treehopper. One study out of British Columbia has looked at, so they put treehoppers on grapes to acquire the virus for 3 days, and then they put the treehoppers in this tube with a sucrose solution. The idea being that they would feed on that sucrose solution and transmit the virus to it, and then they tested the sucrose solution for GRBV. It's just a little simpler way of doing it than using a whole plant or even a leaf. And they did find some evidence for transmission using buffalo treehoppers, so 15%, 9 out of 61. So kind of things we're working on or want to work on in the future is, one is just kind of an ecological question, resolve the species composition of this genus in vineyards in Ontario. So this is, Bisonia is the species that probably 90% or more of the individuals we've collected are. That's what we've mainly identified. This species, Diceros, is not that hard to identify morphologically. It's different color. There's some differences about its features that are easy. And we know we have some of this. I just lumped them all together into the genus for this talk. But there are some other species like Taurina, and there's a few others that are green and look like Bisonia, so they are hard. But with some barcoding, you should be able to determine what other species we have and how abundant they are. The other thing that's kind of, we like to work on is, can we rear the species in the lab like alfalfa treehopper? This would give us the ability to do assays any time of the year almost. But I think the difference between these two is alfalfa treehopper is probably easily continuously reared. There's no diapause, and it lays its eggs on bean plants, and the nymphs develop on those plants. Whereas with buffalo treehopper, it's likely that another host plant is needed for egg laying, and it's probably a woody plant, a tree, a shrub, a bush. And we have not been able to identify that yet, and it's probably not grapes. The other thing is it probably has a diapause, and the diapause might be obligate. That basically means the insect has to go through a dormant cold period, which makes rearing in the lab more challenging. So, and thirdly, if we can solve this, or if we can't, we can continue using field collected, but continue to conduct some more assays. Can we find GRBV in the salivary glands of buffalo treehopper? Does it take more days after feeding than with alfalfa treehopper or not? Is the virus acquired by nymphs like alfalfa treehopper? Is it transferable from nymphs to adults? And will it acquire GRBV or transmit it to free-living grapes at different rates? And in terms of this plant hopper species, I've been looking at some of the related plant hoppers that are in the same family, Cixaidae. And there is a species that is found in Europe called Hyalestes obsoletus that is the known vector of Bois Noir disease. And I think this is a disease that is not found in North America. It's not a virus, it's a phytoplasma. But interestingly, this insect will lay its eggs at the base of basically weeds that are either in the vineyard, but more likely outside the vineyard. And these root-feeding nymphs will acquire the phytoplasma from some of these plants. So then the adults emerge, they have the phytoplasma, they will go into the vineyard and occasionally feed on grapes, and they'll transmit the phytoplasma. But the adults are fairly short-lived, so they don't generally feed and transmit it from grape to grape. They might transmit it to one plant that they fed on. So we know what about the whole soil component of this Melanolieris's life cycle? Are these nymphs able to develop on grape roots? Could they be transmitting the virus from the root of one plant to the root of the next if they move that far in the soil? Do they transmit the virus if they acquire it as a nymph to the adult? You know, in BC, those adjacent vines, here I put 90%, but Jose actually said I think it was pretty much all of them. You know, all those newly infected vines are right next or right beside old vines, so there seems to be, you know, the spread isn't kind of random within a vineyard. At least in BC, it's slowly spreading from one spot. So that would give some evidence to a spread in the roots or in the soil.
Can I ask?
That's it. That's all I got. So in a second. Just to acknowledge these people, Srabani, a former PhD student, technicians, other students and people at AFC, and folks at OMAFRA, as well as at CCOVI and Brock, and specifically SUD. And thank you to participating growers, as well as the different funding agencies. And thank you to all of you and everybody listening online. Okay, go for it.
Did you say it was a slow spread from previously infected vines to newly infected vines?
Yeah, it seems, yeah, like
the tree hopper, do they fly very far?
alfalfa tree hopper is known to be able to fly fairly long distances. You know, I can't, like hundreds of meters, maybe not kilometers. A lot of the literature in California says they don't see much spread like from one vineyard block to the next. It's always more within a vineyard.
Almost like a fortifying sort of around the vineyard, something to kind of follow up with your idea of it being soil borne. Maybe that's.
Yeah, so if the infected vine, I mean, GRBV can be found in the roots of these plants, I'm pretty sure, right?
It is.
It is. So if there was something feeding on the roots, there's potential that it could acquire it in the soil. And then as long as that nymph of that insect can pass it to the adult, if that adult emerges and then flies and already has it, it could then spread it to a different vine. Or the nymph could spread it through feeding on two different vines if one was infected and one was not.
The virus transmissible through grass.
That's a good question. I doubt it.
Yeah, it would be.
I've never read anything about that.
So what? Grass. They're dead bodies.
Frass usually refers to.
I don't think so. Most of these
No.
viruses are obligate pathogens, something that is developed, right?
But I wonder if you tested the frass from a GRBV positive insect, would the frass be positive?
Right, exactly.
The frass necessarily wouldn't pass it to another plant.
But maybe it would be some sort of like your chain of events.
Yeah, it would be a way of getting a false positive in some of these tests if you accidentally detected GRBV in the frass. But I don't think there's any transmission through frass.
I saw the slow transmission with your idea of the soil, and on top of that, the insect being pretty alive.
Yeah. With the Melanolieris, the plant hopper, I have no idea how mobile that is compared to the tree hoppers.
Right.
It can fly, but I have no idea how far. Yeah. Thanks.
Do you know how many generations of this Melanolieris?
I think both of these tree hoppers, Melanolieris, my impression is they're like one generation per year. But in all the traps with Melanolieris, there seems to be kind of two peaks. So I'm wondering if that really is two peaks, or I don't know, we're missing it, trapping it for a couple of weeks for whatever reasons. Weather, could be anything.
The reason I ask is, especially viruses like Grapevine Red Blotch, which is a deployment virus, we always find very low titers for detected through the molecular methods like PCR in low titers in the early season.
Right.
And virus slowly starts to build up in initial leaves, and then not so much in the end leaves. So if the insect has two generations, maybe the later generation have more potential to, more chances of transmitting disease than early season.
Yeah, that one graph where we kind of had two peaks. I mean, the percent positives of those melanolarians for each peak were kind of, more or less the same. There was no, plenty of them were positive for the virus that we captured earlier in the summer too. So I was thinking along the same lines, but maybe it doesn't necessarily work out that way. Yeah.
Does having the virus affect the little bug's survival?
Yeah, that's a good question. I have, from working with it, I'm not sure, but from the literature, I don't think so. I haven't read or heard anything that it would. I mean, just like, well, maybe not just like, but I think, from the virus's perspective, you wouldn't want to impair the insect to a great extent, otherwise your virus is not going to be transmitted to the next plant. I guess a little bit like COVID, right? So, yeah.
Is there a plan at all made to look at roots, to look at see like them
Yeah.
See if it's like
Yeah. So. Yeah, I just put in a proposal for funding to do some of that research. Yeah, we'll have to. The melanolarians are very hard to capture and find, especially live ones to work with out.
Is it a good thing, maybe?
Which is a good thing if it is transmitting the virus, but to work, the buffalo treehopper are really easy to find. They're really robust. They're really easy to work with. Those plant hoppers are smaller, they're finicky, they're hard to find, they die really easily. So yes, we're trying to get the funding and develop some way to study that, but it will be a challenge.
We do have some questions
Sure.
from people joining us online as well. So Josh Atkins is asking if the level or amount of virus within a vine changed the ability of tests to detect virus. If so, is this accounted for when determining spread by potential?
The answer to the first question is yes. Sue could probably answer in more detail than I could. The second question is, yeah, I think like with our transmission assay, like with that excise leaf, there's evidence that those tree hoppers that were positive fed on it. And perhaps, even though we didn't detect the virus in their salivary glands or in that leaf, perhaps it was at such a low titer, such a low amount after only 4 days that we just didn't detect it. We can do more sensitive PCR tests like ddPCR or even qPCR. So, it's possible getting false negatives just because the titer of GRBV is so low in leaves and maybe even in insects.
And we have a couple of questions from Wendy McFadden-Smith.
Oh, thanks. Thanks, Wendy.
Her first question is, was melanolarians found in the Dow block with a significant increase in GRBV?
Yes, but I don't think that was a site where it was. There were a few sites where there were more melanolarians, and I don't think that was one of the sites. But it wasn't one of the sites where it was zero. It was kind of like we found a couple there, but not many.
Okay. The next question were symptoms of tree hopper feeding present in vineyards? So
I have only seen symptoms of tree hopper feeding in 1 or 2 vineyards, and those are organic vineyards, I believe. I didn't show this data today, but we have done some lab tests where we sprayed insecticides on buffalo tree hoppers, insecticides that are registered in grapes for leaf hoppers, and almost all of those insecticides kill these tree hoppers within hours. So in a conventional vineyard, if these insecticides are being used for our other leaf hopper pests, they're likely also killing any or almost all of the tree hoppers that are visiting the vineyard. If they spraying consistently with these insecticides. So, with our sticky cards, we hardly found any buffalo tree hopper. Well, that could be they just don't like to go into grapes, but it also could be that a lot of them got killed if that grower had sprayed with an insecticide, or more than even some growers are probably making multiple applications for leafhoppers.
Our next question is, do tree hoppers typically feed on leaf blade or petiole?
I would say the petiole. Yep.
And then she also asked, how long does it take from inoculation until titer is high enough to detect GRBV?
I can't answer that. But, I mean, it's probably somewhat, what I alluded to before with how you're testing. I can't say off the bat like how titer relates to detectability exactly.
And she had one last question. What does feeding by Milano vineyards look like on Grapevine?
No idea. Yeah, I've never like seen a live one on a grape plant. Even when we've had them on grape plants in the lab, I don't see any type of feeding damage. I mean, certainly in our ongoing work with Melanoliris, that is something that we will hopefully try to answer. All right.
Can I ask one last question, sorry?
Of course.
With the publishing, why haven't you
So we know, so yeah, we know it will feed on legumes, like it feeds on beans pretty well. Faba beans, we keep it on sometimes in the greenhouse or outside in cages. But it will not lay its eggs on those plants. There's different plants that it necessarily feeds on versus the plants that it wants to lay its eggs on.
And you say beans on the patio, so like beans are kind of stocky, is it like stocky plants then, or is there some sort of take on that?
I'm not totally sure about like the structure of the plant and how that relates to like what it wants to feed on or not. My guess is it has a fairly large like number of plants that it can feed on. It can feed on grapes, it can feed on beans. Like there are probably all different types of plants out there that it will feed on. Probably not grasses though. Like I don't think it will feed on grass, but anything else could be very handy. Like we put it on alfalfa because it will feed on alfalfa too.
You're calling
But
being a world transition virus, so they feed on the world.
yeah. Yeah, go ahead.
The Dow that was heavily infected, was there localized spread that you were seeing? Was it more
Yeah,
like it did sectional literature for BC?
I don't know if Wendy or whoever else has collected that data has looked exactly at like where the spread was occurring related to the older vines. I've wondered myself the same, but that is something that we can
are
kind of
looking at root and soil-based vectors. Like, how would you go about identifying the species and then the relative positivity of the virus in those infant forms? Like, how would you go about identifying that?
I mean, in terms of the species, we'd only work with that one genus, and we think it's all one species, so that's, we focus our efforts there. In terms of designing the experiments, I mean, conceivably we have a great plant that is GRBV positive, and we can take root samples and get a positive GRBV test from the roots, and we would, first we want to see if the adults, if we put them on those plants, will they lay their eggs even on grapes? Like they may not. They may want to lay, we may need to try to find some other plants that they are egg-laying hosts. Same as with the tree hopper. But then hopefully, say if they do lay on grapes and we can get the nymphs to develop and they're feeding, can we dig up the nymphs and test them? Are they GRBV positive? And carry the experiment further, and if they're going to feed, hopefully eventually at some point they do emerge and we catch the adults and we can test the adults. That's kind of in a nutshell what we would try, I think, first in the greenhouse or lab. Sure.
Have you found across any research, it's going to be pretty out there, GRB infected insects being eaten and that being to another insect and that
well, I don't think no. I mean, to find the natural enemies of these insects would be interesting as like a biological control approach.
Then see if they
No.
infected. Also interesting.
No, it's not going to happen. No, it's got to.
That's pretty
Yeah, no. It's not going to work like that. With the alfalfa tree hopper, some people have been looking at what it's eating, like which plants it's eating, by looking at what's in its gut and doing that with molecular methods. So we could do that with these as well. All right, that's it. Okay.
Well, thank you so much, Justin. Here's a little something on behalf
wow,
of
thank
me.
you.
And for those of you joining us online and in person, thank you for joining us. And as Justin alluded to, next week we have Dr. Jose Ramon Urbez Torres joining us from BC, and his presentation will focus on sustainable control of grapevine trunk viruses. So we hope to see you then. Thank you.